Hello, I'm Dr Jennifer Ashworth and I'm an Anne McLaren Research Fellow working at the University of Nottingham Biodiscovery Institute. So in my research, I'm interested in the surroundings of cells in the body. So cells are not alone, they're surrounded by a complex network of proteins and sugars. And so when we grow cells in the lab, we want to try and recreate this as closely as possible. And at the moment, a lot of researchers are using a substance called Matrigel, which is derived from a mouse, which is pretty good for growing the cells, but it's not very similar to what we see in the human body. So what my research does is look at creating artificial mimics of the protein composition that we really see in humans. So I work on breast cancer, and so we've looked at breast tissue from human patients to understand what makes that protein network really specific to human breast. And then we can create an artificial version of this network, so that when we're growing the cells in the lab, we're making them behave in as close a way as possible to how they would behave in the human body. And that's really important for being able to test things like new drugs and how the disease develops, so that we can create better ways of testing it in patients. So if we use animal-derived materials or animal models, animal experiments, that's actually very different to what happens in a human. So scientifically, we need non-animal derived approaches and non-animal research to get much closer to replicating how cells will behave in the human body. And what we're after in my research is to almost trick those breast cells that we're growing in the lab into behaving as if they're still in the human body. And that way we can design better treatments and we can understand much better how those patients are going to respond in the clinic. Animal-free research is really important because when we're trying to make cells behave in the same way as they would do in a patient, using animal products or animal models is just not good enough. So that's why in my research, I'm trying to move away as much as possible from anything animal-derived and replace it with really tailor-made systems, tailor-made hydrogels for growing cells in 3 dimensions that are much closer to what we see in the human body.